So, hello everybody, welcome to our second webinar. My name is Miguel Garcia. I'm the lucky guy that managed the core facility at EPFL in Lausanne, Switzerland. I'm very happy today because I will host for this webinar George and Charlotte from Seed Biosciences that will present us a wonderful new device that I'm happy to have in my facility and to work with since almost 1 year in the pre-development phase, and now in this final, let's say, final new device installed with the latest innovation. I'm really happy to have both of you here to present this new device. Let me just introduce a little bit you guys to our audience. So George has a PhD in bioengineering from Professor Barondon's lab at EPFL, and is the CEO and co-founder of Seed Biosciences. George is an opportunity seeker and a relationship builder and a great communicator. He works from some biomedical startup before creating Seed Biosciences. George is received really several awards from Young Entrepreneur as Venture Leader, Venture Kick, Venture in 2018, and we are working together since his EPFL time on different project sorting and as well as acquisition in flow cytometry, and also on this wonderful project, George, since almost 2 years now. Charlotte is currently the business development manager at Seed Biosciences. She earned his bachelor in life sciences technology and his master in bioengineering at EPFL. She moved to USA for his master's thesis in bioengineering at UC Berkeley College of Bioengineering. Still in the US, she was in charge of promoting the exchange of ideas, talents, and technology between Switzerland and North America. As project manager, entrepreneurship and innovation for SwissNext in Boston. After this US experience, let's say, she returns home and starts a new adventure now with Seeds and George. So again, it's really a great pleasure for me to introduce you. We are really happy to have you and really looking for this webinar. For our audience, make sure if you have any question to use the little button that you see just on the right side with the QR function and put all your questions there. So Charlotte will moderate this part. She will take all the questions along the webinar and ask them during or at the end, depending on how George wants to be interrupted during this presentation. On this wonderful COVID situation now, George is in the facility in front of the device. The idea is to have a presentation, but as well have a little demo from the instrument. We're gonna switch from George's computer to the DispenCell computer, and also a little camera that shows to you the device. So our apologize a little bit in advance if we have some problems during the connections. We have tested just before. Everything should go smoothly, but in case, stay online. I really encourage you to ask all your questions during the webinar. This webinar is recorded, and I will provide to all our audience the link afterwards if you would like to see everything more in details at the end. If you have any questions related to the core, to the DispenSoft, please feel free to contact us. So with that, I'm done. George and Charlotte, the floor is yours.
Okay, thank you very much, Miguel. First of all, on behalf of the team of Seed Bioscience, I want to thank you, Miguel. Thank you a lot, you and your team, for putting in place such a nice webinar. I know this is not your first time. You're doing that for other startups in the field, and on behalf of the community, thanks a lot for offering us such a nice, a window to the community and to the field of flow cytometry. So thanks, big thank you to you, Miguel. Okay, so Miguel, can you confirm that you can see my slide?
Yes, yes, it's okay.
Thank you. So as Miguel mentioned, today we are going to have the presentation of DispenCell, the product of Seed Bioscience, in two parts. The first part, I will give you a short introduction to the technology, the product, and how it works and what are the benefits. And then we thought that it would actually be nice that you can see the machine working. So I have it just next to me. So in the second part, we will move to a practical demonstration of the solution. So before I start, I want to give you a quick introduction to Seed Bioscience. So in very short, Seed Biosciences is a spin-off from the EPFL, the Swiss Institute of Technology here in Switzerland. The company is a rather young company. It was created less than 2 years ago. We have used some public money to develop the prototype, and now we just delivered our first product. And this product is basically based on more than 5 years of research here at the EPFL. And at the beginning of the year, we managed to sold our very first product, the very first robots. And actually, I realized that this slide is not up to date because since then we managed to sell two more products despite the COVID crisis. And for that, I'm very proud of my team. Talking about the team, this is just to show you that we are a very nice team of young people with different backgrounds from the business, biology, engineering. And my message here is that we are here for you. So whenever you have questions or you want to have a demo or you are already our clients, we are here to help you at any time. So please feel free to contact us. So today the talk is about single cells. I'm not going to do a long introduction about single cells because I'm sure that if you are here, you know about the importance of single cells in the fields of biology and precision medicine. But just to make an overview, at least that's our understanding, we believe that surrounding the application of precision medicine, there's basically different challenges, whether it is in research, diagnostic, or therapeutic. When you basically deal with this kind of application, you often have samples that are very patient-specific. Very often, when you work with such applications, you have access to a very limited number of cells, sometimes even less than 1,000 or 500 cells, and those cells are very precious. And in this kind of application, the need to reduce the time and costs of the procedures is very important. And as we are moving to clinical therapeutical applications, obviously the compliance of the protocol to the FDA regulation is also very important. So this is something we can discuss at the end, but among you, you might have different applications of interest. The ones we have in mind when we developed this machine are the trendy applications such as CRISPR-Cas9, liquid biopsy, antibody screening, and cell therapy. So in order to address this kind of application, we have developed a very innovative pipetting system for single cell isolation. You will have a demo, like I said before, later, but briefly, this is kind of a revolution in the field of pipetting because this is the very first pipetting system that enables you to isolate single cell one at a time. So it's the one-click, one-cell system. Very briefly, but you will see it later, it's a very compact and simple system for flexible and fast, easy handling. We also provide traceable isolation of the single cell, which is very key to perform a single cell quality control. And last but not least, the system is also capable of handling a very small number of cells. And that's critical when you work with a precious cell sample. So this is here on the image on the left, you have a image of the pipetting system with the tip, the pipette tip on it. On the image, the graphic on the right, you see the system and how it works. So basically, it dispenses a single cell. Then we can detect a signal, which is based on the electrical impedance. And whenever we have a peak, we know that we have dispensed a single cell. So here, one peak, one cell in the well. And when here is a case where you have two cells sticking together, a doublet, we can detect two closed peak, which are the signature of a doublet. So this is basically the principle of this DispenCell robot. So like I said, it works with a pipetting system in combination with a disposable pipette tip, which you can see on the left. So we can discuss later the composition of the sensor, but basically we have a very tiny sensor at the very end of the pipette tip, which is capable of detecting every single cell passing through this sensor. So the pipette tip has a nice gold electrode on the surface, and as you can see on the image on the right, the microscopic view of the sensor is basically based on a Coulter counter, which consists of a membrane with a tiny aperture at the center where the cells actually go through. And basically, when they go through this hole, they generate this peak, which can be recorded detected by the pipette. Maybe in terms of application, the most important information here for you is that this disposable tip comes as a sterile, so you can use it, and you can use a new one every time you start with a new cell type or new population. So this is fully disposable. You don't need, like it is for some of the FACS, for example, you don't need to clean the machine. So here on this slide, I just wanted to show you the way the pipette works. So if we start from the left, the first step, it really works like a pipette. So the first step, you have to, the system aspirates the cells into the tip. When this is done, the robot moves to the first well where you want to dispense a single cell. And then it starts dispensing liquid out. And as soon as the first peak is detected, so the cell, then the flow is turned off, and the pipette goes to the next well, and so on. So this is how the automated single cell dispensing works. So it's basically based on a feedback interaction between the peak detection and the pump, the flow control. Then at the end of the experiment, the system delivers an immediate single cell validation report, which you can see here. So this is like a bit like the battleship. You have in that case, that's experiment number 2. The well plate format was a 96 well plate. And I think it's quite obvious. Basically, you can have validation of your single cell experiment when you have, for example, in A1, you have a tick. That means that you have a single cell into your well, or the growing clone, for example. And in A6, this well shall be discarded, most likely because there is more than one cell. So as you can see here again, that's, we believe, one of the core value of the system. You can have immediate single cell quality control. And this is because we have the raw data. So it's basically an interpretation of the raw data. So in C7, we have in the raw data for this particular well, we have this peak, the one peak signature. So we know there is one single cell with very high resolution. And for example, in E6, here we see the two peaks, which is the signature of a doublet, which is why this well should be discarded. Now, in terms of the overall performance, if we compare that to the existing single cell dispensers on the market, well, first of all, as we said, we wanted to develop a system that is very gentle with the cells. This is why we are using a very low pressure, 1 to 2 orders of magnitude compared to the competition. We can have 2 plate holders, 96 or 384, different shapes, V shapes, round shape, etc. The system can detect multiple parameters on the cells, such as size and doublets. Obviously, we're not using an optical sensor, so we cannot detect fluorescence. However, one of the key advantages is that we can work with a very low number of cells as an input. So if you come with a sample that has 500, 200, 100 cells, we can still handle it with DispenCell. Then one of the other advantage, like I said, it's all based on disposable. So the time for sample preparation, calibration, and cleaning is very short. You don't need to clean the machine, etc. So basically, you can preserve your cell by minimizing the time of the experiment. Regarding the time of dispensing, now, if we just focus on the rapidity of the machine to dispense a 96 well plate, today, DispenCell takes about 10 minutes to dispense a 96 well plate, which is a bit higher than the dispensing time of other single-cell dispensers. However, like I said, the overall process, including calibration, cleaning, and sample prep, is very short. So in the next phase, I wanted to share with you some scientific validation that we performed. So first of all, we did an experiment where we wanted to assess the efficiency and accuracy of the system. So for that, we used CHO DG44 cells as a model. This is, for those who don't know, this is CHO cells are Chinese hamster ovarian cells. And these are the models of choice for the industry, which is why we have chosen that. Those cells were stained with calcein AM, which is a fluorescent dye. And then we dispense the cells using the DispenCell in 96 well plates in triplicates. And then the plates were checked under the microscope by 2 independent operators. So as you can see on the graph, which is the correlation between the DispenCell and the microscope, we have a very high number of single cell in the plates. It took about 11 minutes to fill 196 well plates. The filling rate, so the number of wells with actually one peak signature was 64%. And the assurance of monoclonality was more than 95%. Then we performed a test, and this test was done to assess the impact of the system on the functionality, not only the viability, but the functionality of the cells. And for that, we use a very nice transplantation assay that was developed to assess the capacity of hair follicle stem cells to form hair follicles in vivo. This, by the way, is a result that was published in the peer-reviewed journal SLAS technology, and I'm happy to provide you with the link. But basically, in this test, we took cells from the whisker of a rat, a green rat. Then this whisker was dissociated. And then we used the pipette to dispense the cells individually into wells. So then the cells were cloned, expanded. And then we managed to get 7 clones out of those 33 single cells. 5 of the clones were expanded and transplanted onto the back of a actinic mouse. And then, as you can see on this nice picture, you can see the nude mouse on this picture. Then you see new hair follicles growing. And those hair follicles were actually generated thanks to the hair follicle stem cells that we transplanted. So this is a rather complex model, but study. But I think this shows very well the power of DispenCell to manipulate such fragile stem cells. Then we performed a test with human squamous cell carcinoma cells, which we used pretty much the same procedure. We first dispensed the cells, we cloned them, and then we expanded the clones. And the cells were, each of the clones were then injected subcutaneously into nude mice and led for development for more than 100 days. And I think here I don't need to speak about that. The image showed the result very well. We were able to get growing tumors on the back of those mice, which was showing again that the DispenCell had no impact on the property of tumor cells. So with that, should we move to the testimonials? So I will let Charlotte speak about the testimonial while I am preparing for the demo.
Hi everyone, it's nice to have you here on this webinar today. So yes, like you heard, George is preparing and setting on the demo for just in a few minutes. And in the meantime, I'm going to speak a little bit about testimonials from our first customers. So here we have Martin from the Laboratory of Integrative Systems Physiology here at EPFL. He is working with skeletal muscle cells. So this is a very typical case of customer that we have. So using a really little amount of cells, fragile cells, and actually a sample that is quite rare. So where we were able to help here is that Martin wanted to do some single cell clones. So DispenCell was gentle enough and precise enough to be able to dispense single cells gently and very precisely. And the good thing is that he was able to actually track the actual number of suspended cells per well. Another case is here, the case of Dr. Marian and Dr. Johannes from Laboratory of Systems Biology and Genetics here at EPFL also. So here's a little video on how we were able to help in their experiment.
Hi there, I'm Marian.
Hi, I'm Johannes.
We're coming from the Planck lab, and here at our lab, together with our colleagues, we developed a no-cell left-behind platform for single-cell RNA transcriptomics called DISCO. So one of the challenges that we faced during the benchmarking of our system was when we tried to accurately quantify efficiency of our system, and we wanted to sort 100 cells using independent method and then quantify how much we get out with our system. However, this turned out to be an impossible job because most of our experiments at best we were getting 6 cells out before we could even start sorting them in our system. That's where DispenSoft helped us out because it allowed us to accurately quantify and retrieve retrieve almost all cells before we even starting to benchmark our.
So as you heard, the issue here was that Marian and Johannes had to sort a precise number of cells, so 100 cells. That is something that can be a little challenging for FACS, as like the number of cells is really low. Here, I think we could be like a really nice complement to FACS, helping sort that precise number of cells in a really small sample. So I think this is a really nice value of DispenCell. We were able to help them be precise enough by sorting the correct number of cells they were expecting, and by that helping them benchmark properly their platform. So yeah, those are quite typical cases here that we encounter every day. I think George is probably ready now. So here we go for the most interesting part. I think it's how the device works, and George is going to show you all of that right now.
Okay, so Miguel, can you hear me?
Yes, we can hear you,
well.
and we can hear also your device.
Okay, really good. That's good, which means that it's working. Can you see the software?
Yeah, we see the software, and we have the device on the little pop-up window that appears on the screen.
So you're able to see my fingers here, right?
Yeah, exactly.
With the robot. Okay, very good, very good. So if anybody, if someone is not able to see both at the same time the software and the robot, please raise your hand, and Charlotte will help you make sure that you can see both of the image. This is really important, that you are able to see the robots and see the software at the same time. Okay, so let's go. So what you have here on the table, this is the baby. This is DispenCell, the brand new product, which was delivered early this year, and which is now available, commercially available. So this pipetting robot comes with a computer as well as a kit. In this kit, you have a couple of components. The first one is obviously the most important one. It's the dispense tip. That's basically the magic tip for dispensing. So this is the pipette tip with the gold electrode. Then here we have another pipette tip, very classic pipette tip, but with a right bore orifice. I will explain why it is important, but it comes in the kit. So we have the two tips together. Then we have other goodies. The first one is a cell strainer, which you can buy on the market, but we decided to include that into the kit. And this is important, like for every application in flow cytometry. It's important, obviously, to filter the cells before the experiment, to remove the big clumps and the big aggregates of cells in order to avoid clogging of the tip. And then we have a washing unit you will see later in the protocol why this is important. It's basically a plastic reservoir, which you put here. And then we have another bag, sterile pouch, which contains methyl cellulose-based medium, which is a solution that we have optimized to increase the performance of the dispensing. And it basically consists of a medium based on methyl cellulose, so it's a fully biocompatible material. And it's basically just more viscous than water. And with that, with this viscous solution where you have to put the cells, you basically have a higher dispensing efficiency. So this is the kit. And then obviously you need to have plates, well plates. So you can use standard plates like this one. This is a 96 well plate, which you have to pre-fill with medium. In that case, it's a demo that simulates a cloning experiment. So we put cell culture medium inside, but then you can use different type of plates, like 384 well plates, such as, for example, when you want to do a single cell RNA-seq, you can just dispense your buffer of interest directly into the plate. Okay, so now let's have a look at the interface. Here on the software, I have basically two buttons. The first one is view the results. We will see that later. And run the assay. So I can just click on run the assay. Then the system initializes. It takes a few seconds. Okay. So when this is done, at that step, you have to install the tip directly on the pipette. So just take this little device. You see it's made of plastic and it's sterile, so you don't need to, don't touch the tip like that. Just hold the plastic device, then hold the pipette, open the part here, and then you just, like it's standard pipette, you just plug the tip onto the rod. You see, very simple, and then you just put back the pipette. In order to make your life easier, at every step of the process, we have a page like this one, which explains to you how to do the procedure. If you're not sure how to do it, even though you've read the user manual and you still don't know how to do it, to make your life easier, we have put videos like here. Just click on the link and you see now I have a nice YouTube video which shows you how to do it. And by the way, one of the nice features of this interface that you can see actually on the left, you see all the steps that are presented, and basically the software is taking you one after the other. So when this step is done, you just click on done, next. Here, obviously, that's just a little reminder that you have to have the alignment of the gold electrode on the front. Just next. Here is a step of alignment. You can just align the pipettes. We have a small pin here where you have to align pin to pin. When this is done, click on next. Okay, here are just a few reminders. You have to label your plates, very important. Here is a step where you have to do the sample preparation. Like I said, it takes about 10 minutes. And this step, basically what you have to do, you have to take your cells, then put them at a certain concentration. It's about 10 to the 4 cells per milliliter. And you have to pipette your cells directly into this optimized medium. Again, if you're not sure how to do it, just click on help, and you have a very nice video which shows you how to make this cell sample preparation. Click on next. Here you have a few reminders just to make sure that you're doing the things correctly. Did you filter your cells, filter down, mixing down, and no bubbles? Okay, this is very specific to the sample preparation, but again, this system is made in a way that we avoid mistakes. So you have to check every box. Then here you have to place the wash, which you have to fill with PBS or medium. You just put it here, click on next. Here is a message to make sure that you have removed the leads. You'll be surprised, but sometimes people forget. So we put a message, a check. Okay, here you have to place your place at the right placement. Okay, remove cover. Plate orientation, A1 left corner. Then here is the step where I forgot to put the tube here, my bad. But I put the tube here with the cells. And here is the step where you can do the loading of your sample. So far, it's you have to set a time, but very soon we will have you will be able to set a volume. So for demonstration, let's put 5 seconds. And I just click on load. So then the robot moves. Then it goes inside the cells. And now it is aspirating the cells inside the tube. Okay, so now we have the cells inside. In order to remove any cells that could stick on the surface of the tip, we have implemented a very nice step of washing. I will do just one cycle for the demonstration, but you can do. We recommend usually to do 3. And basically, the tip is moving into the wash, which contains PBS, and is just doing a mechanical wash of the tip. It takes a few seconds. Okay, for this step. So now we are moving at this part, which is the step where you have to select your plates. Here you can do doc 1 or doc 2. You see on the software it's showing you the 2 docs in question. You can do 96 or 384 well plates, and you can choose the well shape format as well. So here we have a 96 well plates. Here, if you want, you can change the name of your plate ID so that it matches the label of your plates. Then I click on next, and here is the page where you have to set the detection parameters. So here I can set the number of cells that I want to have in every well. So it can be 1 if you want to do single cell experiments, or 10 or 50 or 5, whatever, it's up to you. Here is where you can set the number of cells you want in every of the wells. And here is the parameter of interest. That's basically the threshold in ohm, which is the electrical unit, the impedance unit. And that's basically the threshold that enables you to make a discrimination between the debris and the cells. And 300 ohm corresponds to a cell of 10 micrometer. And here is basically a timeout. If nothing happens after 30 seconds, then the robot will move to the next one. So for demonstration today, I'm not using cells. It's just a simulation, so I will just put a timeout of 1 second. And at that stage, basically, you are almost ready to go. Here, that's where you have to select the wells you want to dispense. So I can choose to dispense half of the plates or the entire plates. That's up to me, depending on the experiments that I want to do. And that you can do for the 2 plates, doc 1 and doc 2. So let's just do a simulation with the 6 first wells. Okay, and here I just click on dispense, so that's the one click. Then the robot goes into the well, and it will dispense the single cell. So again, like I explained before, it's just the robot is pushing liquid out, and as soon as it detects the first well, then the robot moves to the next well. Okay, so here I put a timeout of 1 second, but usually, like I said, on average, the system takes about 10 minutes to do one plate, which is about 5 seconds per well. Okay, so now we're good. Basically, I can take my plate and put it back into the incubator, and you're ready to go home at the end of the day. And let's say now the day after, you want to check the single cell quality control and make sure you have single cells in your plate. This is something you can do very quickly. So I will just click here and restart the app. And now I will click on view results. Okay. And here you see all the experiments that Miguel and his colleagues have done here at the facility. So I will just click on one experiment, for example, this one. And here you see the layout of the plates. And I will click on A7. Now I have a window that pops up. And here is actually the full traceability of the experiments in terms of impedance signal. So what do I have on this graph? So on this graph, well, first of all, you see the label of the file. This is number A7 for the plates in question, run number 2. And this graph is showing the impedance signal in time. So here I have the time. And here on the y-axis, I have the impedance in ohm. And the impedance is the blue signal, which is basically, we have a variation between, let's say, 30,000 up to 33,000. And now I will show you, I will try to explain, but I think at the end you will see it's rather simple. I will do with you the quality control. So what we have on this interface, you see the yellow labels, that's actually the instructions given by the robot. So here I have a label that says start enter into the well. So we have rather high impedance, and then all of a sudden the impedance decreases. And this is because this decrease here is because the tip has entered into the well. And then I have another label which is enter into the well and send command for pressure control. And here the pump is switch on. So now at that stage, after 412 seconds, we have some liquid that is being dispensed, right? And then we reach a plateau. And then all of a sudden, after 450 seconds, the impedance increases and goes up. And what you see here, and this is the things of interest for you, you see the green label, which is pointing to a peak. So I will just use this tool and zoom here, and what you can see is your very nice single cell. So this is your single cell. And if I go back.
Sorry to interrupt you, George. We don't see the little pop-up window that is supposed to appear and where we see the impedance peak. At least not on the screen.
Okay. Okay. I spoke perfectly. I'm sorry. Let me see. Share screen. Okay. Can you see the screen now?
Yeah, it's better.
Okay. I'm sorry. I didn't realize. I'm afraid I have to start from the beginning. So again, in this window, what you see is the impedance signal. On the y-axis is the time. On the left axis is the impedance. And then in blue, that's the impedance signal, which is recorded on the pipette tip. So we can see all the steps of the dispensing in the well A7. In that case, it's well A7. So when the impedance decreases, that's when the tip, enter into the well, into the liquid. Then we start dispensing some liquid out of the tip. And here I will just zoom. You see on the green label here, it's pointing to a peak. I will just zoom on it. And here you can see the very nice peak here, which is the signature of the single cell. So now I just go back to this data and can zoom here. And because you don't see any other peaks like this one, this is basically the proof that in this well, we have dispensed only one single cell. Okay. So just want to show you another example. So now you should be able to see the interface. That's very good. We click on A9, for example. Okay, so I actually clicked on A11. So I have to start again, share with you this window that you should see now. That's very good. And here, let's zoom. So you basically see the exact same profile, high impedance, then a sharp decrease, then a plateau here. And if I zoom, I just zoom here. If I zoom, I think you will all be able to tell me how many cells we have in this well. Miguel, how many cells do we have in this well?
2.
We have 2? Yes, that's correct. So you can zoom on each of the cells. Okay, here one cell, and that's my second cell right here. Okay, so in that case, because the peaks are well separated, this is not a doublet. These are actually 2 cells that were dispensed at the same time in the well. So in some cases, it's possible that you have 2 cells in the well, but you can always detect and discard the well using this quality control. So I think I am done for the demo. Okay. So Miguel, should we move to the question part, question and answer?
Yes.
Okay, so I suggest maybe Charlotte can take the questions.
So sure, we had a couple interesting questions on the forum. I think it was nice to actually share all together. So Stella had a really interesting question that was, what is the minimum and maximum cell size?
Okay, Stella. So the minimum and maximum cell size is determined by the size of the aperture in this pipette tip. So today we have clients that use mammalian cells that have cells ranging from 5 to 20-25 micrometer, and this can be done with this tip. Now, if you want to dispense bigger cells, let's say 35, 40, or 50 micrometer, we will just make new tips dedicated to this range of size. The same is true for smaller particles. But again, as of today, most of the clients are coming with cells that have a diameter between 5 to 25 to 30 micrometers.
Also, a question from Stella was, can we dispense bacteria like E. coli?
Okay, Stella. So yeah, I think I answered your question. It would be a matter of adjusting the aperture of the tip. For bacteria, though, with the current system, we are not capable of dispensing bacteria. Bacteria are too small. I'm not saying it won't be possible in the future, but with the first product it's not possible.
So we had a question from Roberto about if it is possible to perform single-cell analysis from scrapped small amount of tissue, like scrapping a small amount of tissue, cells out of a small amount of tissue.
So, like I said, it's possible to run DispenCell when you have a sample with less than 500 cells, 100 cells. Now, is it possible to do it from scrapped, like raw tissue? I'm not sure. I guess it really depends on your dissociation protocol, whether you're able to dissociate the cells properly, most likely using trypsin EDTA or I forgot the name of the enzyme that is used for that, but of course here the sample preparation is important. Basically, we need to make sure that the cells are already in single cell suspension. The pipette is not capable of dissociating a tissue. They need to come as single cell in the sample.
And another question was, do we have any more users using single-cell RNA? Since it's also a question from Stella saying that it's also a problem with doublets.
Yes, so I would say today half of our clients are using DispenCell for cloning application, and the other half is using DispenCell for single-cell RNA-seq. So yes, it's possible to run such a protocol. As of today, we don't have published results, but it will be
Sorry, George, we don't hear you really well. We have like an echo. Can you repeat your answer, please? Probably more in front of the mic.
Miguel is asking if you can repeat more in front of the mic.
Miguel, I think I'm just going to go back to my computer. Okay, Miguel, can you hear me?
Yeah, but I think the computer from Charlotte is close to you. That's probably the reason that, yeah.
Turn off the sound. Okay.
Yeah, better.
It's much better, right? Okay, sorry, I was confused. So I just want to go back to the question of single-cell RNA-seq. So like I said, half of our users are doing this kind of experiments, and we are working with them for to have a publication together and be able to share the data. As of today, if you have particular question regarding single-cell RNA-seq, I think I can just put you in contact, direct contact with the clients if you want to address specific questions regarding this protocol. And regarding the particular question of the doublet, DispenCell is very sensitive and is capable of detecting doublets and discard the doublets from your sample. And this is clearly well validated. We have a nice detection efficiency of doublets. Charlotte, do we have more questions?
I don't think we have for the moment, but if needed, we can answer you by email or by, anytime.
Okay, maybe for the last part, Miguel, you want to take the lead?
Yep, and come back. Okay, I think you see me again. So thank you, George and Charlotte, for this wonderful presentation. It was really, I think, interesting to see the device working in live and also to hear the little noise generating by the equipment. Looks stronger that it is really in live. With the mic we have this amplification, but really in the lab it's not so noisy, especially compared to our equipments. Yeah, again, really thank you for this this little presentation, the demo, doing the demo live. It's not, especially by Zoom, it's not really the ideal condition. So thank you to you guys to make that effort for our audience. So for the rest of the audience, feel free to reach out directly to George and to Seed Biosciences if you are interested to test the device or to have more information about the DispenCell. I'm sure they will be more than happy to answer all your questions and to discuss directly with you guys about our special experience that you will probably to do with the DispenCell. For our for the EPFL users, we are happy to have the DispenCell in our facility, so please don't hesitate to contact us directly if you would like to have more information or to see the device physically installed somewhere and to test it with your own cells. If you have some protocols to run, please feel free to contact us directly. We will be more than happy to help you. We are on close contact, of course, with Seed's team in order to also develop some new protocols. So if something needs to be, let's say, optimized, we have George and his team ready to help us. So please don't hesitate to contact us. And if you pass by the facility, you will see the device directly installed on your left side when you come inside the facility. With all of this, I would like to thank you again, guys, for this wonderful presentation and to make that effort to do this this demo in live. To our audience, thank you to join us today on this device. Feel free. Yeah.
Excuse me, but can I ask you a question?
For sure.
Because, like you said, we've been working together for some times now. You have a machine, and when you approached us, actually, you came with a problem that we did not realize. You came with this the need, a particular application where you wanted to use DispenCell downstream of fax.
Exactly.
In the first place, we thought it would be actually two instruments entering into competition, but with you, we realized that it's not the case. Can you just give us a few words about this nice complementary workflow?
Exactly, George. And it's fun to discuss about that because, yeah, in the beginning you start to be like a competition with fax sorting, and for me, it's not really the case. For me, it's more a perfect complement to fax sorting. So, as probably you know, it's not so complicated for us to dispense and to clone your cells directly on a 96 well plate. So as soon as you come to us and to sort your cells on a plate, we are able to perform it. My question is more related with the discussion that we had with your users, is when they come with this traditional question, are you sure that you have a cell inside of my well? So I can guarantee that, and probably you know, that we are not sorting cells. What we are sorting with the sorter is droplets. So we can guarantee that theoretically we have a cell inside of the droplet, and we do our best to sort that droplet that contains the cell that falls inside your well. And we have techniques or protocols to validate that the droplet falls directly inside the well, and we can fill the 96 well with droplets. But I think the easiest way to really be sure that you have a cell, it's DispenCell. And when I discussed the first time with George about the device and all the things that the device can do, for me, it was really evident or obvious that could be an excellent complement, because we have more and more users that are interesting to do genomic or afterwards. So come to us with really a few amount of cells on a subpopulation, and they would like to after go to the genomic facility to run a 96 well plate. And probably all of you know how expensive are the reagents for genomics, and it's really important for them to be sure that when they put the reagents inside the 96 well, that in each well they have a cell. And we cannot guarantee that with our machine. We can test it. We can show you that we are really put a droplet inside each well, but we cannot guarantee that you have a cell inside of your droplet. So our idea is more now to sort in a bulk, and this allowed us to sort more than one population at the time, and after bring this small amount of cells to the DispenCell, and to dispense the cell directly on a 96 well on a more gentle way also. And with the software that George showed you, we have a clear way to identify the wells that are empty and the wells that contain 1 or 2 cells. And inside that software, we can also export this information and bring it directly to the facility that goes this downstream application. In our case was the genomic facility, and only dispense let's say only dispense the consumables for the genomic inside the wells that you would like to have something, because you can show that we only have a cell. So yes, this was the first, for me, the first really important application where I include the DispenCell in the workflow, and we have more and more people now requesting to do RNA-seq and genomics application. They bring us a small amount of cells, and after they go to this kind of application. And I think DispenCell is really, for me, a piece of that puzzle, a piece of this workflow, and starts with the sample preparation. After it comes to the sort, where we are able to sort this small amount of cells on a bulk way, on a small alicot, and after goes directly to the DispenCell, and have at the end a plate where you can really guarantee that in that well you have a cell, and on that well you don't have a cell. And this is not only for genomic, if you, of course, after do cloning application and different other application, and you need to grow a little bit your cells before really dispense, of course, with this device is the perfect device to do it. So in our facility, I see the DispenCell has a perfect complement to our different applications and different services, than more than a competitor for our equipment, let's say. Did I answer to your question, George?
Very, very well. Thank you very much, Miguel.
You're welcome. You're welcome. So for our users, for the users that have never tested DispenCell with us, and they are interested in doing this kind of application, of course, feel free to reach out to me or to my team. We'll be more than happy to help you and to discuss a little bit more in details all the protocol.
Good. So fax and DispenCell, the great combo.
Of course, and I think on this on this new, let's say, new reality, especially with this interesting virus that bore us since a few months. They have other kind of application that could be developed with the DispenCell, where the DispenCell could be included, especially on sorting, when you are interested to sort this kind of infected cells, and you need to really to guarantee some security, some safety, some biosafety. DispenCell could be really interesting to be combined with different other kind of sorting technology or sorting approach. That's for sure will be interesting and more in the future, for sure. Did you have more question, George, for me, or if the audience have more questions to Sid's team, please feel free to use the QR option there. If it's not the case, let's give them 1 or 2 more don't say seconds, but more minutes to try to answer question. If it's not the case, once more, please feel free to contact DispenCell Seed about DispenCell directly if you are interested in this device, or to test the device directly with your cells. They will be more than happy to provide you with a demo and provide you with more information, and especially with codes, and George. And for our users, as I told you, we have the device in our facility, so we'll be more than happy to help you and to show you directly like a live demo with people from the facility. How the DispenCell could be interesting for your cells and in your applications. So no questions, what I see actually, no, nothing is popping up on the QR. So once more, Charlotte and George, thank you really, thank you very much really for take the time to show us this new device, and especially for the demo. And Zoom is not always the most ideal, and especially to switch from a computer to another one. Actually, they are in our facility using one of our device. So thank you so much to establish that and to spend some time to test it. For our audience, thank you so much to join us. I remind that this webinar was recorded, and I will provide the link to the record as soon as my computer is agreed to convert the video, and I will publish it also in our YouTube channel, a new YouTube channel where you have our 2 webinars. With all that, thank you so much, guys, for this wonderful presentation, and I hope to see you soon. And I hope to see all our audience, especially from EPFL, soon around or across the corridors in our core facility. Take care of you. Enjoy the rest of the week. It's not so sunny. Sunshine, actually, it's more raining, but good opportunity to back to work on safe conditions. Take care of you. See you soon. Bye-bye, and thank you, guys.
Thank you a lot. Bye-bye.